Okay, many of our dead branches will support a wealth of different fungi that are producing fruiting bodies. And what we're seeing here is an example of one of the most common wood decay fungi we have in our flora here in North Carolina and actually in the southeast. This is an oak branch that fell. And as you can see, there is an awful lot of this yellowish orange fruiting body of a wood decay fungus growing along nearly the entire length of this particular branch. This is referred to, or these are commonly referred to as the parchment fungi. In some cases, a shelf fungi, simply because of the top bending over that you see there. These can be produced and often are in great abundance, as you see here. The spore-bearing area is on the underside of the small cap or top, and this is where we find the spores that are produced and will be wind blown away. This particular fungus here is very important in nutrient recycling and the breakdown of woody plant parts such as this branch of this particular oak tree. If we were collecting this, we would take a portion of what you see there, carefully removing it, and writing down on our information sheet the type of tree it was on, especially if you can get it down to something like an oak rather than just maybe a deciduous tree, because a lot of these wood decay fungi turn out to be fairly host specific. There are certain ones that occur only on conifers such as pine and fir in our western part of our state. Others that occur only on hardwoods. This one, for example, occurs primarily on oak, although we have it reported on a number of other hardwood or deciduous tree species. We would then fill out the information sheet or field sheet and put it in with the collection. Oftentimes for the collecting of mushroom-like fungi, of wood decay fungi on wood, we will put these in a paper bag rather than in a plastic, or rather into wax paper. It really doesn't make much difference when it comes down to that. When you're collecting wood decay fungi like this, one wants to make sure that you get a good sample of it. In some cases, you'll find that the reproductive body is fairly large in some spots and fairly small in another. I always like to collect both the large ones and the small ones, simply because often the very large reproductive bodies will have already expulsed their spores. And if spores are needed eventually in identification of these, you simply won't have it. Whereas if you collect some of the smaller reproductive structures, fruiting bodies, they will almost invariably have some spores being produced on them, so you can get those to look at under the microscope to aid you in identification.